Okay, welcome back, everyone. I'd like to introduce Dr Belmonte from North Carolina State University, who's going to tell us a little bit about how to do intercell signaling and use that to control the cell cycle. He's also going to show us a little bit about how to load other kinds of subcellular models into.
Okay, hello everyone. So as James said, I'm Julie Belmonte. I'm a former PhD student from James Glacier, and it's my pleasure to talk to you a little bit about how to add other types of models into CompCell. And well, we have already been done this quite extensively using the COVID modeling, but here I'm going to show you another example and walk you through how to do other modes where cells communicate with each other. And so this is the plan for this 1 hour of exercises today. So we're going to talk a little bit about multi-scale modeling, CompCell 3D, and specifically I'll talk about intercellular contact signaling. We're going to build a delta notch model in CompCell 3D, which is a specific type of signaling that goes between cells and helps them pattern many things. Then I'll talk a little bit about SBML models. We're going to go through a couple of exercises, and in the end, we will build a simple colonic grip model in CompCell 3D. And if you do not know what this is, that will be made clear as we go along. So, as you probably know by now, the CompCell 3D is a very flexible modeling environment, and you can do many things with it. You can model many cellular behaviors, like you can specify where it is, the volume of the cells, their shape, their movement, their adhesion. They differentiate into another types of cells. You can even polarize the cells, making elongated and have different subcellular structures that will be covered a little bit later this evening. And you can also make the cells grow and divide and die, and then you can model things like cancer and many developmental processes. But one thing that is not in the scope of the cellular pots modeling or the Glaser Granet Hogweg modeling is what's happening inside the cells in terms of its molecular reaction. Okay, what's going on inside the cell. And you have all these biochemical kinetics happening that dictates what the cells will do, right? And this can also communicate to the next cell, and tell the cells can communicate through these sharing molecules between one another, and that can now regulate many things that's going on at the cell and tissue level. So some examples of biochemical kinetics include the cell cycle. So cells have an internal clock that tells them when to divide. They also have circadian rhythms, cardiac rhythms if you're modeling maybe the heart, and other things like and oscillations. Delta notch patterning, you want to have your model have Wnt pathway, FGF pathway, and that's especially important when you are modeling developmental processes. And the list goes on and on. There are many things that you may want to add. So one example that we usually give to students is how to add self-cycle models, and there is many self-cycle models available in literature that try to explain what regulates when the cells are going to grow or divide. Here we are just adding this self-cycle model inside the comp cell as a reaction kinetics network, and in color you see the levels of, if I'm not mistaken, cyclin B2, and they go up and down, and once they go down, they pass a threshold, then the cells know that it's time for them to divide, and then we implement that into CompCell 3D. The cells see the spike in the signaling, and then they know that they have to undergo mitosis and have division. Okay, so there's a little bit of stochasticity in this model, even though, and you see that initially all the cells start more or less synchronously, but as time goes on, you have some kind of, they start to dephase and undergo division at different times. The other example that we're gonna work on today is the delta notch patterning, where the cells, they initially have the cells communicate with each other in such a way that there is some kind of, they kind of, there's a negative feedback between at the level of one component in one cell and the level of another component in the other cell, and after some kind of transient time, the cells and eventually make up a pattern, which where in this case they have high notch, what you see in red cells, and low notch in the other cells, and you make this kind of patterns, which you see a lot which is used to pattern many things in the body. So the idea here is that besides the actual mechanics of cells movements, growth, and death, you can also add all these other things to the into CompCell, that you can make a very complex model which will have things happening at multiple scales. And you have already seen a little this with the COVID exercise that you had at the first 3 days of this workshop. But here's another example where you can see all of them in action in a context where the cells are growing and making patterns. So this is a multi-scale of somatogenesis, which is the process by which your body grows and subdivides into transient structures called somites. And these are the particles of a vertebra. So here you have a bunch of cells growing at the end, and as they grow, they also diffuse some chemical called FGF signaling, which is just diffused using the diffusion solver, right? So you see this gradient of FGF high here and low at the other end of the growing body. And here on the rightmost panel, you see this very big network of, there actually is 3 negative feedback reaction kinetics network, where the cells are just going up and down in many of its proteins and mRNAs. And so you have which is called a clock. And once the cells are see low level of an FGF gradient, they actually read the current state of the clock and use this information to decide which fate they will eventually have and how they will differentiate. And depending on how they differentiate, they will express different addition proteins that will actually make the pattern or the subdivision of the previous immature tissue into cells that express high levels of this protein or other protein, and then they eventually detach from each other and make these boundaries. So this is an example of a multi-scale model that you can do in CompCell, which actually we are doing using information at the cellular level, how the cells behave, move, and interact with one another. Tissue-wide kind of signaling going on, which is the diffusion of gradients, and intercellular signaling, where each one of those cells has a copy of this gene regulatory network or metabolic reactions next network, and they interact with this other information from the outside, and they set up what the cells eventually will do and respond, and then all the 3 levels are interacting with one another. Okay, so this one kind of thing that you can do in CompCell, and the question now is, okay, so how will you do it? And that's what you cover on today. Okay. So there used to be that there only one way to add reaction kinetics models to CompCell 3D, and the usual path was that you use a developer support, just like Copalzi or Tellurium that you learned in the first week of this course, if you took the first week. And then you can build up our OD model and generate something that's called an SBML file, which has all the description of your system of ODs, the variables, the parameters, and each conditions. And then you can use this SBML file and load up in CompCell software, and then you can associate that to each one of those cells. But now there is also a different way to go about this. You can actually you can skip the developer software and write the model directly into CompCell or in the Tweedit editor, and then use it directly in your model. And that's the way that we're gonna use today. So all this sounds a little bit like old news because you have already seen this in the COVID modeling, but we're gonna go over that again one more time and show how to do from scratch. Okay. So there are multiple ways to add a reaction kinetics network into CompCell. So suppose here you have your simple cell model with two cell types, and on the right you have a reaction kinetics network, and the details of this does not matter. It's just think of this as the system of all these that you want to add to your CompCell simulation. So you can either add each one of these components to all the cells of your simulation, and now all of them will have an individual copy of this. Okay. You can also choose to add this reaction kinetics model to just one of the cells or a selected number of cells in your simulation. You can also add these reaction kinetic models to each cell type in your simulation. In this example, we just add a copy of this reaction kinetics or OD system to all the blue cells in your model. Or you can do, I believe, what you had done for the COVID modeling, just add the reaction kinetics to the CompCell without necessarily associating that to a particular cell in your model. And that's you use to model something that's external to the cells, but is varying over time. And this can be used for PBPK modeling, which is physiologic-based pharmacological kinetics. Okay. So all this can be done from the Tweedit, and this is a list of all the commands that you can use to do each one of those things that I mentioned before. So we start by creating a string here where you actually write all your equations, and here I just have one line for simplicity, where you have a flux J where A goes to B at rate K times AB, and then you can have other rates, and you can also specify what's the initial condition of A, of B, what's the value of K, and you can have many copies of this. So you define a string at the beginning, which is going to be all the reactions that you go on that exist in your OD system. And then you have here the command to add this model to a single cell, to add this model to a single cell, but just listing the cell ID instead of the cell itself. And you can have a list of IDs. You can also add the model to a cell type, as I said before, and this is the command to use it. And you can also add the model to the system without associating to a cell itself. And when you do this, you can have to define exactly which model you are adding. So this string here you can have multiple models running at the same time. And so if you have more than one model defined here, you can specify which one of them you are adding to the cell. You can give a model name for easy access afterwards, and you also have to add the step size, which is going to convert the time units of your model into the time units of the CompCell. So this is all the commands that is used to load the model into CompCell, and here is what you do during simulation. So the very first you have to do is have to make sure that you tell CompCell to run the model. So you have to time step and this we call it SBML model, but is by SBML what you mean is the system of ODs that you are associating to the simulation. And every time you call this function, you are running this OD model for this time step that you define here. Okay. And this step here will have the units of whatever the units it's in your OD model. It can be seconds, can be hours, depending on what your OD model is really modeling. So the other thing that you can do, you can go over the cells in simulation and read the SBL model and read the concentration of a given molecule on your or species in your OD, this A or B, or you can read the parameter. You can read of this K here from your OD model. And what you have to do here is you have to just grab the cell, read the SBL model of this. Here you have to specify the model name, which is going to be this name that you have here. And then you specify here, in this part here, the name of the species or the parameter that is in with your OD system. So here you can just grab the name of this variable or molecule in your OD system. You can also use the same syntax to override the current value of the species or parameter in your model. And in this case, you use the same syntax here, the cell, the SBM model, the model name, and the name of the parameter or species that you want to modify, and then have the equal afterwards, and then you specify the new value of your OD system parameter or species. You can also copy the current state of the OD mod from one cell to the next, and you can also delete an SBM model from a cell if you decide for whatever reason that you don't need to run that OD model inside that cell anymore. So for example, let's say you kill the cell or you inactivate the cell, and then there's no need for you to run the OD model every time in that cell, every time you do this command here. So you can save yourself some time and remove the model from that cell, and then next time you run the simulation or the next Monte Carlo step, that model will not be run inside that cell. The cell will not have that particular reaction kinetics modeling going on there. Okay, so I don't know if there's questions in the chat. I don't have easy access to it right now. So I will count on the other members of the team to look at the chat. So should I go on?
I think so. I think you're fine.
Okay. So I would like to illustrate how this works. We're going to talk about the delta notch patterning. Okay, so if you look at many of the tissues in our organisms, or even in other organisms such as zebrafish, Drosophila, the fruit fly, or ourselves, you see many instances where the tissue itself is colored or in a very particular way that looks like a checkerboard. So here in the zebrafish retina, you see that if you color the photoreceptors in the retinal tissue, you see that some cells are colored and some are not, and you see there are more cells not colored than colored, and it's not really random. It's a very regular pattern where you have a colored cell surrounded by non-colored cell. In this case, the color means that you have a photoreceptor in that cell, but not in the neighboring cells. If you look at the hair cells in the Drosophila inner ear, you see the same kind of pattern. Some cells are activated while the cells around it are not activated, and these are the cells that are actually gonna make up a little hair, grow a hair out of it. That also happens in the same kind of pattern happening in the wing of the Drosophila. And if you look at your intestine, there's this your intestine is made of a bunch of crypts, And at the bottom of the script, you have also this kind of pattern happening where you have some cells colored and some cells non-colored, and I'm being very non-technical here. And these colored cells are the stem cells in the base of the crypt that regenerates your intestine, and these other cells are what's called the pannate cells or the supporting cells, which are the ones that go around it, and that regulates and controls the stem cell niche in your intestine. And then you have many of them in your intestine. In each one of them, you're seeing the base of a crypt, which is this kind of wheel that surrounds them. So that would be the, if you guys are seeing my mouse, that would be the surface, the internal surface of the crypt, and then you have all this the intestine, and there's going to be all the crypts lying around it that renews the crypt and maintains its and regulates its homeostasis. So you have and in this particular model, it's more interesting because the cells are growing and dividing, so the number of cells and the neighborhood of the cells is changing all the time, and yet you have to maintain this kind of structure where you have one cell activated and the other cells non-activated, right? Or vice versa, depending on how exactly the cells will read this kind of signaling pattern. And in all these examples, what's making this pattern is called the delta notch signaling pathway, where you have some kind of, and delta, so this is a pair of receptor and ligand in the surface of the cells, which makes the communication between the two neighboring cells, and then they exchange some signals between them, and the outcome of the signaling process is that some of the cells will be high in delta and some other cells will be high in notch, while being low in delta and low in notch respectively. So this cell here, let's say, is high in delta and low in notch, and these surrounding cells here are going to be high in notch and low in delta. So that's the outcome of this delta notch pattern mechanism that you see in many tissues. So this is just some figures that try to explain exactly what's happening. You have the notch and delta receptor and ligand communicate with each other in such a way that activation of one leads to the downregulation of the other inside the other cell. So as this evolves over time, initially you end up with this pattern, and so there's a negative feedback mechanism going on. So, and the cells eventually, some cells are going to have a slight uptake of delta or notch, and then this gets increased over time. So in this sense, there is a positive feedback inside one cell where one gets a little bit higher notch or delta signaling that downregulates the other process, and they lock in a high state of notch or low state of delta, and vice versa. Okay. So this is more or less a very brief overview. There is no time to go into the molecular details of exactly how this happens. What it matters to us is that someone else in 1996 published a very simple model of how this goes on. So, and this is the model proposed by Collier et al. In 1996, where you have this the delta active, it signals to the notch on the next cell, which leads to the delta inactivation of that cell. And once this delta is inactive, that upregulates the notch on that cell, so this cell becomes high in notch. And once delta, and on the other hand, once delta is active on this cell, and you do not have active notch in this cell, that favors the further activation of delta in this cell. And by having this kind of mechanism, you kind of lock the two cells in having this one high in delta and this one high in notch. Okay, so they propose a very simple OD system to describe the signaling process, where you just have two or maybe two variables or state variables, the delta concentration in the cell and the notch concentration of that cell, and the delta concentration of the cell. Well, for both you have a decay term, right? But there is a cross activation, so the delta it's increase, but is also a function of the number of notch inside that cell, and the notch in the same cell is a function of the amount of delta that it sees from the neighboring cells. So this D here with a bar on top of it is the average delta from the neighbors, and this thing is on the wrong place here. Now it's correct. So N is the notch inside the cell, delta the notch is the delta inside the same cell, and this D with a bar on top of it is the average delta from the neighbors. And when you run this ODE in many cells, and then you have to have this kind of readout from the current cell neighborhood, what you actually get in the end is this kind of pattern emerging, where some cells have high delta and low notch, and some of the cells have high notch and low delta in them. Okay. So we know that now we have a model, this OD model that's very simple and is able to produce exactly what you want to do in our CompCell simulation. So the way that we can implement this is by following more or less this recipe. So our first example, which we're gonna do today, we're gonna implement this model in CompCell. So we need to create a space filled with cells. You're gonna be using the uniform initializer. You're gonna just type this model here using the Antimony language. Then you're gonna add the cell to each of the cells, right? And then we have to run the simulation. And when you run simulation, you cannot just simply run. You have to always update this delta average that the cells see from the other cells. So what we have to do, for each Monte Carlo step, you have to loop over each cell in your simulation, loop over the neighborhood of that cell, and with the amount of delta from these neighbors, calculate its average, and then pass this information back to the OD system, and then update the OD system, and then do it all over at the next Monte Carlo step. Okay? So, and here's the model, here are the parameters, and of course the last thing that you want to do, you want to visualize the patterning that we're getting. So we have to create a visualization field in CompCell3D to actually see the amounts of delta and notch in each one of those cells. Okay, so that's the example that we're gonna do in the next few minutes. I don't know if there's any question in the chat, and if I'm going too fast or too slow.
You seem to be good.
You seem to be good.
The only thing people have asked for is if you could make your screen a bit bigger.
I guess I could, but now I have to make this a double screen now.
It looks okay now.
Looks okay now? Okay.
Yeah.
So are you guys seeing this screen now?
Yes.
Slide and
yeah, we can see your double screen.
And it's tweeted. Okay, excellent. So let's now implement this model. And I'll make this a little bit smaller so you have more working space here. So we are all in tweeted, and the idea here is that we are gonna do this together. So let's make a new CompCell project, and I'll call this my DN model. You can call this whatever you want, right? I'm gonna just call my delta notch model. And let's choose Python plus XML. Hit continue. We can leave the space as this. Maybe I'm gonna make a little bit smaller so we have the assembly is gonna run faster. So we're gonna make it 50 by 50. And one important thing for this simulation is that I want to make a square with periodic boundary conditions. So instead of having no flux on the x and y, I want to have periodic on x and y. So this is a little bit misleading. This is actually for the x and this is actually for the y. And the z parameter, we just leave it 1. We don't want to go into 3D. But in principle, the model can be run in 3 dimensions as well. Okay. So let's type square. Let's leave these numbers for now. And rectangular slab as the initial cell layout. Okay, so 50, 50, periodic, periodic. These are the most important things that you have to change here. If everybody's good, I'm gonna hit continue. And now I have to add a cell type. So we only have one cell type. I'm gonna just call it cell A. So we're gonna type here cell A and hit add here. So do not forget to do this. The cell is only added when it shows up in this list here. Okay. So I type a cell which I call cell A. Now I can go on. So one very important thing is that when you specify a cell name, and that also is valid for the name of your simulation, you cannot start with a number, okay? That's a bad thing. And you cannot have spaces. If you do this, you're gonna narrow, because then once you have your cell type in a simulation, it will interpret this as 2 separate strings, and then you have undergo lots of weird problems. So you can have a number like this, cell 2, but please no spaces and no starting with numbers for your cell names, okay? I don't know if that was mentioned by my check, but it's important to remember, okay? So medium, cell A, we are ready to continue. No chemical fields, we don't want that. Continue. And maybe one contact and volume flex. That will be the only things we'll be using in this simulation. But one thing that we definitely want to use is the cell neighbors. Okay, so this one here. By the way, are you guys seeing this floating window here?
Yes.
Okay, I hope you are. Okay, so because you want to go over the cell neighbors to calculate the average delta in each one of those cells. So we only want these 4 things turned on: contact, volume flex, center of mass, and cell neighbors. So make sure you have all these 4 on, and if you have all of them on, you can now hit continue and configuration complete. So let's click done, and let me double click here, and this is all the files. So now I must ask if this kind of font it's okay for everybody, if I should make it bigger or if I should make it a different color scheme.
No, that looks okay.
So if everybody's okay with it, let's just now make the simulation as we want. So let's go back here to the XML file and make sure we have everything that we wanted. Yeah, it's periodic here and here. We have our one cell type, the import track is on, and here in the uniform initializer, I want this to fill up the whole space. So I want to make my x go from 0 to 50, my y going from 0 to 50, so it fills up the whole space in the simulation. Okay, so you make simulation from 50 to 50 on both directions. You won't have the same, the information initialized to cover the whole thing, so we won't have no holes behind. And let's make our cells width to be 10, just to so we have exactly 5 cells by 5 cells in this space. And this is all the changes we want to make here in the XML part of the code. So you do the changes. This icon here gonna be red because you make change, so you have to make save, right? So make sure you save, and now let's go to the steppable part. Okay. So here in the steppable part.
Sorry, Julio, they asked you to go over the changes you just made one more time.
Yes. So all the changes I made here is make sure that the coordinates of the box spans the whole space of the lattice. So before this was 10 and 40. So what I make is to go from 0 to 50. So it covers the whole space of the lattice. So I want to have a confluent space. With confluent, have also fill up the whole space. So we do this for the X and the Y. So this box min and box max, I don't know if you guys have seen this before. It draws a box inside of your lattice and fill this box with cells. And you have to specify the one corner of the box and the other corner of the box. And it's going to draw it and fill that with cells. And I want to have one corner at X and Y equals 0, and the other corner at X and Y equals 50. And the last thing I did is I make all the my cells to be 10 by 10.
You're not sharing your screen.
Julio, we can't see your screen.
I cannot see my screen. Okay. Can you see it now?
Yes.
Yes, okay. So sorry for that. So these are the 3 places I made changes: 0 to 50, 0 to 50, and here the width of the cells will be 10. So this means that I create a cell that's 10 by 10 pixels. So if this is fine, we can now go to the steppable file, where we're actually going to define the steppables. Okay. So here, the first thing we want to do is define the string, model string. We're going to type this OD model here, and we just type here model string and then make triple quotes. Right, and here you're gonna write so maybe instead of work on here live in the interest of time, maybe I should just show how this is done. Okay, so the first thing, you have to type this equation here. So you define a model string where you actually define your delta notch model, and you have the first reaction here. So you write something that goes into the delta notch, and this is going to be the rate at which. So I'm just here typing this part here, and I think this color scheme does not make it very nice to see this, so I'm going to change this. I think this is more visible. So to type this into as a model string in your CompCell, you have to write here your first reaction. Something goes to delta, and this is exactly how you type this kind of equation here. Then you have to type the second reaction. So second reaction, the rate of change of notch inside the cell, which is going to be given by the average to the k divided by a plus the average to the k, and this is the k term for the notch variable. And here is where we define the parameters. We have v equals to 1, b equals to 100, h equals to h and k equals to 2, and a equals to 0.01. Okay. And this is the initial conditions for the delta, the notch, and we have to say something for the average. And let's say that everything is 0.4, 0.5, and 0.5. So this is how you define your model. You just type this directly into the tweedit, and you save as a variable called model string. So then in the Steppable, you have to add this line, add antimony to cell types. So if you do not know how to do this, you can just go here and use the CC3D Python, and you go to SBML solver and add, there's an option here that's called add antimony model to cell types. You click here and automatically paste the command for you. So here you can set the file. You don't really need that, so you can delete, and then you have to add the model string, as I did here, model string. Model name, here I gave the model name VN. The cell type, in this case I add to my cell A, that we define when you use the wizard. And the last thing that we need is set up the step size, and here I just give a step size of 0.2. When you use the drop down menu here to add to use this command, it also asks for the initial conditions. We're gonna not use this today. We're gonna only use this these four commands here. The string, which this thing here where you wrote up the model, the step size, the model name, which I called DN, and to which cell type I'm adding the model. In this case, we only have one cell type, so all the cells of type cell A will receive a copy of this model. So I'll delete this. And I don't want all the cells to start with exact the same initial conditions. Otherwise, there will never be a break of symmetry in your system. What I want to do is actually assign random numbers for the initial delta and notch concentrations. And I want to assign them just the way that the people that wrote this paper did, which is they add random numbers between 0.9 and 1 for the delta and the notch at each one of those cells. So to do this, you do a loop over all the cells in your simulation, and you change and for each one of the cells we have a copy of this model. So you cell.spml, and then you're accessing all the SPM models or the rational kinetic models that you can have inside the cell. Then you have to specify the model name here. So this dn here should be the same as this dn here that you have here. If I had to specify delta notch like this, I would have to use delta notch here. But so that's why it's preferable to use a shorter name, so you do not waste too much space. So here I'm accessing this model, and then I have to specify which variable or parameter I want to access from my model. In this case, I'm accessing the D, which is going to be variable here, and I'm assigning a random number between 0.9 and 1. And in this case, I'm using NumPy, so we can do import NumPy as np. I don't know if this was covered in the Python module that you had before, but NumPy is a collection of mathematical functions that you can use in Python. So I can import this NumPy library. And one of the things that it does generate random numbers for us. So I can just, by this using np random and uniform, I generate a uniform random number between those two values. So here I'll generate a uniform number between 0.9 and 1. So all the delta and notch for each one of those cells will have a random number between 0.9 and 1. So all this is done in the start function of the Steppable. Okay. So that's just setting up the initial conditions of the cells. So now for each Monte Carlo step, what you have to do is have to time step the SBML as we have here, but you also have to go make a loop over each one of the cells and then each one of those neighbors and calculate the average delta of the neighbor, so you can update this variable here, which is this variable here, in the model. So to do this, we do a loop over all the cells to simulation, and we're gonna calculate set up these dimming variables, the delta average equals to 0 and the number of neighbors for that cell to be 0, and then we do the loop over all the cells. So if you guys don't remember, to do the loop over all the cells, you just type this here into Intercell 3D Python, visit all cells, and we do the loop for you. Once you are inside the loop, you can do the loop over all the cell neighbors, and the way to do this, you go again using the Intercell 3D Python drop-down menu, go to visit and select the option visit all the cell neighbors, and when you select this, you automatically get all this code paste for you. And in this case, you have here is grabbing all the neighbors of the cell, and here you are getting the neighbor and the common surface area of the neighbor. You just saw that a few minutes ago with TJ. And the first thing you want to do is you check if your neighbor is a cell or a medium. So if you just do if neighbor, it returns true if it's a cell, and returns false if it happens to be the medium. So in this case, it will always return true because we have no medium in the simulation. All the space is filled up with cells. But still, it's good to have this checked. Okay. So you grab, for this particular cell, we grab all its neighbors, and then we access the neighbor SPML and this delta. And to do this, you do exactly as we did here in line 36. You get the neighbor.spml.delta notch, and then we grab the data from that cell. So you store a copy here, and for those of you who do not know, this plus equal sign here is just means it's just the same thing as the average equals the average plus this new value. So this plus equal just means that you are adding into this value whatever you have on the right side of your equation. So this and this they are different syntaxes, but they do the same thing. The average is your old average plus this new number that you put in there. So I'm gonna remove this. So you calculate you're storing all the delta values in this dummy variable here, and at the same time you are counting the number of cells that this particular cell has. Once this loop over all the neighbors is over, if the cell has enough neighbors, at least one neighbor, then you calculate the average self. You just divide the total delta that the cell sees from the neighbors by the number of neighbors that the cell has, and you store this value in as the new value of the delta average in the delta notch model. So again, you do a loop of all the cells. For each cell, you do a loop of all the neighbors with the delta concentration of these neighbors, make this average, and then add this information back into the ODE model. So once you do this for all the cells, then you can time step the simulation. And if you do not know how to write this, you can just again go to CC3D Python menu, go to the SML solver, and time step SBM models. You do this, and then we're going to just paste the command for you. So this is everything that you have to write to have your delta notch model running in your CompCell. And the last thing that you may want to do is you want to create a new class or a new Steppable to visualize the delta and notch concentration on each one of those cells. Not sure if this was covered, but the way to do this, you just go here on the Python delta notch Steppables, click here, double click, right click here, and there's option called Add Steppable. So again, you go here into this window here, right next to, usually on the left side of it, you open your delta notch model, you go to where you have the Python script, right click, and then there's this option Add Steppable. Once you click this, it's going to open this menu here where you can ask you to add a Steppable name, and in this case you can write whatever you want. Let's say delta notch watcher if you wish. How frequent you're going to call this Steppable, and in this case every Monte Carlo step. Let's leave it as a generic Steppable, and you can already choose if you want to visualize, so have some kind of visualization Steppable, and you can choose this to be a scalar associated to each cell. So you can already choose this one. And when you click this, you automatically add a new Steppable to your simulation, which will be which will have already a scalar field to it. So the way I did this here is I have two scalar fields, one to visualize notch, the other to visualize delta, and the only thing that I add is I add this parameter variable here, which is called self n equals to this and self d equals to this. So when CompCell creates a scalar field for you, it just creates like this. I find it very helpful to also have this added in front of it, so I can store and it'll be easier to access it later. So of course you're going to create just one for you. If you want to create a second one, you can again go to CompCell 3D menu Python and go under extra fields and select the scalar field cell level and select the option to create. And this is already telling where it should be created. It should be created in the init under the init function of the Steppable. So you click that, I'm gonna paste this for you, and then you can just write field 2, and my suggestions always have this as an easy access to later. So you do this and you create one for notch, the other for delta. In this case, I make one self.n and self.d. So I'm not sure if this was covered, but all the variables that you create inside the Steppable that have the self dot at the beginning, will be available in all the functions in your Steppable. Okay, so you create those two, and at each multicolor step, you first clear this field, okay, and then you're gonna populate this field with actually the values of the notch and the delta in each one of those cells. So here, to populate this, you just write self.n. So this you are the field here that you just created, and then you have to have a open square brackets and specify which cell you're writing this to, and then here I'm grabbing the notch value from the delta notch model and assign it to this field. So here I again created the notch and delta visualization fields, I clear them, and then for each cell I associate the notch field for each cell with the actual notch value that the cell has. And then here we are grabbing the delta notch model, the notch from the delta notch model for that cell. So as you can see here, this command can be used in any class that you have. You do not have to have this visualization necessarily inside the Steppable where you do this. You can do this separately in another Steppable. Okay, let's see if one person thinks too fast. Most people think it's okay. So if you type all of this, and you can either type this on your own, tweet it as we go along, or you can grab this model. There's already a solution for that in our shared model folder. So if you go here to CompCell 3D course, Google Drive, student materials, and Julio's lecture examples, all this is coded here in this delta notch.zip file. You can download it and just download the computer and run it, and it should run fine. Or you can do together with me and type everything here. So this is everything that we did. I'm gonna give you a little bit time to copy this. And once you do this, you save the model, and you can run that in CompCell, and you should have something that looks like this. So let me go here, switch screens. better, I'm gonna just go back here, left click here, opening player. And this is the initial condition. You would see a cell field with all the cells filling up the entire space, and you should have access to the notch and the delta. So let's look at the notch here and the delta here, right? And if I just hit play, you see that it seems that for a short period of time nothing seems to happen, but eventually some cells become high in notch, like these ones here, and some cells is low in delta. So here, those cells here are high in delta, and the other ones are low, and these cells here are high in notch, and the other cells are low in notch. So you have the reproduction of this pattern mechanism when you run this model, and I hope that also happen in your computers. Okay.
Could you repeat just to show them how to add a steppable using Twitter?
Yes. So I'm gonna stop simulation, go back to here. So if you want to add, so in principle this can be done inside the same steppable if you wish, but if you want to add a new steppable, you just go here, open the model that you are working on. It's going to be either this one or this one. You find the Python module, which we have to go the one that has steppables, right? Then you right click on top of it, and there is this option, which is the third one, which has add steppable. This only works if you click on the file that has steppables on it. If you try to do this here, it's not going to work. If you try to do the XML, it will not work. You have to click on the one that has steppables in its name. Right. Add steppable, and then you specify, you give the name. And in this case, you want to add a scalar cell level field, so you can already check this, and then when this generates the steppable, it already generates with a scalar field inside it. Is it okay? So I do not want to add a new one here.
Okay, I think that's fine, Julie.
Okay. So if you run the model, you should be able to see these two options, notch here and delta here. If you want to have two windows, you can just click here and this button here, and it's going to generate a new window to visualize the cells. Okay, and then you can have one look at the notch, the other look at delta. You hit play, and you should be able to see something that looks very similar to what I have here. So if you have this working, some exercise that you can do, well, you should have this, is to increase the level of memory fluctuations or the temperature of the system to see how the pattern changes. You can maybe, so this I'll suggest it because we will be running out of time today, make delta signaling proportional to the fraction of shared membrane area. So instead of, so here if a cell just barely touches the next cell, it has the same weight on the average as the other one. What you can do with this is making, instead of having like this, you just multiply by the common surface area and see if there's any difference. You can also make a loop over the neighbors of the neighbors and grab the delta average of these also neighbors and calculate this average of a higher neighborhood of the cell and see if this how that will change the pattern. Maybe you have more separation between the dots, right? And you can also run simulation with different cell arrangements, have cells on a line or something like this, and see how the pattern looks like. So these are exercises that I suggest you guys to do either with your own version of the code or if just grabbing the delta notch version of the code that I shared to you. So I'm not seeing the So was everybody able to do this? So on yes. So I'm checking the pooling. So most people are done or working on it. Okay. Well, by the exercise, I think what you mean is get the basic model working. These ones here, we suggest that you do on your own after the workshop or between today and tomorrow. So the people working on it, do you need more time to complete the exercise or should we go on so I can just show you the next slides? I'm not sure how to proceed now. This took a little bit longer than the projected 1 hour.
I think we need to move on so that we can get to the compartmental cells.
Okay. Okay, so what I plan to have shown next that you can also do the same thing by saving the delta notch model as an SML file, right? And I have a couple slides here that you can check, guys, later to see what SML file looks like. So this is a basic reaction. That's how it looks like when you look at inside the SML file, and you can load that into CompCell using basically a same set of commands as before, right? It's just that on the drop-down menu of CompCell, we'll be using a different set of options. Instead of having Antimony, you have the SPML, but apart from the loading of the model and the fact that you have to specify exactly where your model is in your computer, everything else looks exactly the same. And on the class files here, I have the same exact model using the SPML file instead of the Antimony. And if you look, open it, you see that it looks exactly the same, except that you don't have that model specification here. You just refer to a file, but everything else is exactly the same. Okay. And the other thing that I was planning to do is make this kind of simulation where instead of have the whole simulation fill up with cells, you just start with 2 cells and also periodic boundary conditions, and you have the cells growing and dividing, but you do the delta notch pattern on top of it as it goes. So if you implement that, and that has to be an exercise for you guys to do on your own, and here you look at the levels of notch. As the cells grow, the cells have a new neighborhood, so the pattern that you had before gets short circuit and there's a new pattern that emerges. And eventually, when you have a confluent of the whole system, you end up back to a new steady state. Then if you do this and get something that looks like this, the next thing that you can do is repeat the simulation again, but now have only the notch positive cells growing. So in this case, only the cells that are high notch, so you have to make a look. If grab the notch of that cell, if it's higher than, let's say, 0.9, you allow them to grow. If not, you do not allow them to grow, so that's going to be a different version of this. And the last thing that I will ask you guys to do, if you have more time, you do the simulation again, but only allow the cells that are at the center of the simulation do the pattern and the growth. And when you do all these kind of rules in your simulation, start from a single bunch of cells, you're basically kind of almost at the full of, you are very close to have a full model of the colonic crypt, which is this kind of system that you've seen before. So in the system, we have those cells here at the bottom of the crypt that are growing and dividing and generating the crypt. These cells undergo a delta notch patterning, but this delta notch pattern is just restrict to the bottom of the crypt, and only the cells that are reaching notch receive a steam sulfate, which they are the only ones that grow and divide. So you can implement this model in CompCell. I'm just going very quickly over it. You can do that. And one way to, but instead of using this three-dimensional model of the crypt, you can just simplify this kind of cylinder shape and unroll it, and have a model that is periodic boundary condition on this direction and not on this. Fill up with cells and say that you're going to restrict the delta notch signaling only to the cells at the bottom, and only the cells at the bottom that are reaching notch will proliferate. And as the cells grow and divide and push the whole tissue up, here you can remove the cells by adding a check if the cell center of mass is above some certain value, and then you just set this target volume to 0 and then remove the cells. And that would be a very simplistic colonic crypt model of how the colonic crypt works. In reality, that's lots of wind signaling regulating the size of the crypt, and then they're being secreted by some of those cells here. But that will more or less give the same kind of results. And you can either do this as your own. If you have no good model to do in the hackathon, you can maybe try to do this one and make it more complicated to reproduce the real system. Or you can just, again, grab this already coded crypt file that I have here in the student materials. And if you want to take a sneak peek of how this looks like, again, I have the exact same delta notch model being specified here. It's just that I have the same visualization, everything here is the same. The only difference that I have a check here to see where my cell is at the model, and only do this if the cell is there. And here I add a few steppables to grow the cell if the cell is reaching notch and below a certain level here. I grow them. If not, I don't do nothing. And if the cell on the y direction, the vertical axis is higher than 150, I just kill the cell by setting a target volume. And there is also a mitosis steppable class where you go over all the cells, and if the volume double, we just divide the cell at a random orientation. Okay? And this command here, self-clone parent to cell, automatically takes care of cloning a copy of your SBML model from the parent cell to the child cell. Okay, so this model here, I go here very quickly. You can find a copy on the student materials folder. You can open it up, take a look at yourself, and try to run in your CompCell player. I have to, yes. And here if I run this model, oops. No, this is the wrong model. Let me close the player, go to edit, ask to play it again. Okay, so this is the model running. Here is seeing delta, here is seeing notch, here is seeing the cell field. And if you just download this simulation to your machine, you should be able to see exactly this going on. You have delta notch signaling kept here at the bottom of the crypt below 40, and the cell notch rich cells grow and divide, as you can see here. Of course, as soon as you have new cells, the delta notch patterning changes, and this pushes the cells upwards, and once they pass this threshold, I just remove the cells from the system. Okay, so this is a very simple model of the clonic crypt that you can do as an exercise by yourselves. You should do all the tools to do it, or you can just download the model here at the folder and play with the model yourself. And now in the interest of time and keep things moving, I'm gonna stop here the